We can see your screen. Thanks so much, William.
Thank you so much, Sarah. So tonight's presentation will focus on three main branches of image processing here at OPALS. And for those of you who do not know, I'm a returning intern for OPALS, so I went through the very same learning process that many of you are about to go through. So once you get the hang of it, I promise it's not that bad, but you just need to get through that initial learning curve so that you can get on your way to image processing. Tonight, we'll focus on three main platforms through which we do our image processing. We'll start with MATLAB, then move on to Python CellPose. I actually have a little demo prepared for Python CellPose, so hopefully that's a bit exciting. And finally, Fiji ImageJ, which is probably the most ubiquitous platform that we use here at OPALS. Okay, so let's dive right into MATLAB. MATLAB is first and foremost a programming language that was developed by MathWorks, and it was developed side by side with Simulink. So MATLAB is kind of the more computer science side of this program, and Simulink is in charge of creating models that cohere and adhere to the MATLAB language. So MATLAB is especially helpful because it is used primarily by scientists and engineers. Can I see how many of you guys, and raise your hands if you're interested in STEM careers? Please raise your hands if you are interested in STEM careers. So almost everyone I see are basically interested in STEM careers. MATLAB will be incredibly useful to all of you, no matter what field of science you go into, because as we march deeper on into the 21st century, we are going to head deeper into the information era. And the main enzyme of catalysis for the information era is, of course, computer science. If you're completely new to computer science, don't worry at all, because UC San Diego actually gives free access to all OPALS interns for you to learn and get used to MATLAB. Personally, when I joined OPALS last summer, I was not at all a computer science guy. I had maybe taken an introductory course in Python, but of course that didn't help at all with MATLAB because the two are actually fundamentally different in terms of the coding language. Either way, I highly encourage you guys to review Dr. Linda's earlier email. I think it was sent just this past week about how to download the MATLAB software. And if you haven't already, please do that because some of your projects will involve the use of MATLAB. What kinds of projects will involve MATLAB, you may ask? I actually wanted to present a case study of how MATLAB is used in OPALS projects. So this is from OPALS Group 7 during the summer. I was not part of OPALS Group 7, but I'll try to give my limited understanding of their project through my explanation. Or if anyone here is from Group 7 during the summer, if you guys would like to explain that instead, though I'm not sure if anyone from Group 7 is here. Okay, so my understanding of Group 7's project is that they were studying PC12 cells. So PC12 cells are a line of cells derived from the adrenal glands of mice. So adrenal glands are the organs that sit above your kidneys. And PC12 cells are very interesting because they have varying thickness that the team tried to measure. And the way they did this was that they laser ablated the PC12 cells. What that means is that they're basically using very high power lasers and slicing these cells so that these cells experience damage, and thus the cells might have some molecular mechanism to facilitate repair and modify their thickness based on the varying strengths of the lasers. And this model that you see here, that looks like a topographical map, that was developed with the help of MATLAB. So you know how ubiquitous MATLAB can be and how we might use it in the lab in your future career in the OPALS internship as well. I will also say that even if your project does not really involve MATLAB, actually a majority of projects during the summer, I think, did not have to use MATLAB. We still highly encourage that you guys get a good foundation in MATLAB because this will serve you well in the future. Specifically, MATLAB is very useful for image analysis, especially when you're trying to quantify cell structures or track cell movement, but those are just a few of the uses. And here is where I get a little bit of bragging time, and that is to say that last summer in the OPALS internship, we actually had a little tournament between each of the groups to determine who could basically finish the most amount of MATLAB certificates during the internship. My group, Group 18, decided to be pretty competitive, and we teamed up with Group 17. Each person in our two groups averaged around 15 or 16 MATLAB certificates by the end of the program, which brought us up to a total of more than 150 certificates. So that is the record if anyone is seeking to beat it this year. But just in summary, MATLAB is very useful in various fields of science. They provide a lot of different learning opportunities through these courses that you'll get access to once you download MATLAB with the UCSD license, and you can get started on learning how to use MATLAB, even if you are completely unfamiliar, like I was, to computer science. We'll now go into Python CellPose. So CellPose is a program that was developed by a web-based community that is particularly passionate about cell biology. And it's very helpful because it is a deep learning tool for analyzing biology images. What are we doing here for the most part in OPALS internships? We are most likely going to be doing some image analysis. What's also very interesting about CellPose is that you're able to train models on image sets. I actually have an image set that a previous mentor provided to me to practice how to use Python CellPose, and I'm sure if you guys wanted to contact either him or you could contact me for some practice, I could also seek to provide you with that set of images. But the basic idea behind Python CellPose is that during the summer, a few groups used CellPose to help quantify the amount of cells in a given image. I will say that Python CellPose comes downloaded with some certain base models, though those models are not very helpful and you'll have to train them to your own use. And this is particularly helpful if anyone in your group knows Python. If not, that's also okay. So I also prepared a little bit of an activity before we get into the CellPose demo. First of all, I want to ask everyone to count the cells in this image. For this purpose, let's just assume that each cell is one blue dot. And you probably can provide me an answer in the chat within 20 to 30 seconds. You could say, oh, this is easy. I can just count them 1, 2, 3, 4, so on and so forth. But the problem with that is that while some images are comparatively easier to analyze, others may look like this. And this is the problem with manually counting cells or manually analyzing images. It's that if we can find ways to computerize and to quote unquote mechanize the program by using a computer science algorithm, makes life easier for both the interns, the advisors, the mentors involved. So this is where we dive into the CellPose workspace. So I'm going to stop my screen share for now. I will make a quick detour to paste two quick links in the chat. The first link is a link to an introduction to Python provided by our previous mentor, Connor Lee. So I will make this available to everyone. And if you guys are complete beginners, I think this will provide a good intro to Python, because that's also the document that I used to get started with a bit of the coding that I wasn't familiar with at the beginning of the internship. I would also like to provide everyone the opportunity to get started with Cellpose and basically the downloading process. So I will go ahead and paste the second link into the chat. This link was actually a set of instructions that a fellow intern and I wrote up during the summer, because our group was having a lot more trouble than we had expected trying to download Cellpose onto our Macs. So I'm sorry if you guys aren't on Macs, but most of our team members were. But even if you don't figure out how to download Cellpose yourself on your computer, I'm sure ChatGPT can help you figure that out. Okay, so let's say you followed all of the steps in learning some basic Python and then going through with the downloading process of Cellpose. I'm going to go ahead and share my screen once again. Okay, give me just a quick second to share my screen. I might just share my desktop. Okay, so I'm gonna go first to Anaconda Navigator, because this is the program that I use to launch Cellpose. So once again, Cellpose is entirely dependent on you having Python or a navigator to access the program. So for me, I have Cellpose already downloaded. So you guys can, when this presentation ends, you guys can figure out how to download Cellpose yourself. But I'm just going to show you what Cellpose might look like, which is taking a tiny bit of time for now. In the meantime, does anyone have any questions? That was a good amount of information. Does anyone have questions? Feel free to raise your hand and ask straight away, or put your question into the chat. I'll go ahead and start the initialization with Cellpose, and then if you guys have questions, please ask. I see a message in the chat. I'm still confused on what we have to download. So it depends on the user. So everyone has different preferences, but I'm assuming, because that I had no experience with computer science before the program, I'll just treat it as if everyone does not have any experience with Python or computer programming. So for accessing Cellpose, you need two things. Number one is you need a Python runner or a Python navigator. So some people will tell you to use VS Code, so that's Visual Studio Code, but personally I prefer Anaconda also on Anaconda. So Anaconda helps you to navigate to terminals, which is needed to open Cellpose. You also need to download Cellpose. So to download Cellpose, you can go through the second link to access the instructions. But hopefully that answered your question, and if not, I can address it later as well. So basically, to initialize Cellpose, because I already have it downloaded, I type in the Cellpose command, and that should do the work of opening the workspace for me, which usually takes a bit of time too. This is what you should see when you open Cellpose. And I'm going to drag an image that we worked with into the, is this the right one? I think so. I'm going to drag an image into the workspace, which allows me to view the image. This isn't necessarily the image that I wanted, so I will take a later field of view, and you'll see that all of these bright spots are kind of surrounding this darker region here. So you'll learn more about your individual projects, but a rundown of what's seen here is that basically we use a very high-powered laser to strike the center of a cell culture. And this laser is kind of like, when it impacts the cell culture, it's kind of like a ripple in a pond if you throw a stone into the pond. The ripples expand outwards, and the bright spots here are actually cells that are either dying or they are upregulating proteins to facilitate cell repair. So basically, what you want to do in cell pose is that you want to be able to train the model. And the first thing that you need to do with a practice set like this is you're going to identify the regions with cells. And that's going to be especially notable if you can see the bright outline here, for example. So what I'm just going to do is I'm going to right click and basically make a circle or an oval around that site. And I'm going to continue drawing these masks, as they call them, around the sites with cells. And once I have drawn enough masks on enough cells and in enough of these images, I can use this to train the model. Personally, I'm not entirely clear on how the process of training the model works, but I am going to show everyone what a trained model looks like. So this was some of my previous work in masking cells. So you can see a lot of these masks, and the central dogma in this is that the more the better. The definition of deep learning and machine learning is that once the computer has picked up some principle of what you're trying to do, you continuously feed it more information that you want it to process, and slowly the machine will pick up on the correct regions. So, for example, let's use this model called Nuclei for William because I was playing around with cell pose during the summer, and let's see how it works. So this is based on the previous work and the masks that I drew, and this is a new image, and let's say I run that model. So what it should do is that it should automatically draw some masks for me and quantify how many cells or cells there are here. So it says 25 regions of interest. So that's how many cells it's picked up on. If I realize that it did not pick up on a few cells in particular, I can go back to the image, I can continue circling cells, and then I can send the pictures back and train the model more. And that's basically how you'll use Python cell pose. I will say that for Python cell pose, you also won't necessarily get to use Python cell pose. My project didn't use Python cell pose, but we were kind of we were experimenting with it at one point, but this is also not something that you quote unquote have to do, but we highly encourage that you have some exposure to all of these different platforms. Now moving on to the last portion of the presentation tonight, and that will be on Fiji ImageJ. So whereas we had Python cell pose and MATLAB, Fiji is much more ubiquitous in our projects. So this is the primary image processing tool that most every group will use. So it's very important for you to get an early start on learning how Fiji ImageJ works. So a cool fact about Fiji ImageJ was that it was developed by the NIH a few decades ago, and the most important functionality that Fiji ImageJ provides us with is that it helps us to view, edit, and annotate different images. And this platform, Fiji ImageJ, is also where you'll be doing most of the manual work unless you can develop an algorithm to do that for you. So for example, this is a case study from the group that we worked most closely with, group 17. So you can see some of their images here. They were identifying regions of calcium buildup after the their cell cultures were hit with laser induced shockwave, which is once again the you throw a stone into the pond analogy. That's what they were looking at. They were also trying to, at some point, determine the epicenter of these laser induced shockwaves, so precisely where the laser hit. So here you can also see the other functionalities of Fiji Image J. It can help you measure distances in pixels. And this is, I will say, a lot of manual work, but that's science. You put in the hard work and you experiment over and over. You don't know if you can be successful, but when you do reach those results, that's one of the best feelings you'll have in your life. And summarized by my fellow intern from Group 17 over the summer, to cook, you have to be cooked. That's an OPALS slogan that will live forever into the future of the OPALS program, and I'm happy it started just over past summer. I'll now go ahead and talk about some of the work we did in my group's project. So actually, I do have a fellow intern tonight who was working on the same project as I was over the summer. So I'll at the end of this slide, I will also call on him to see if he has anything that he would like to say about what the work we did in our project. But the basic rundown of our project was that we were looking at cells after the shockwave occurred. So after you hit the cells with a laser and the shockwave has expanded out and injured or damaged the cells near the site of shockwave. The problem is you need to do some manual work to quantifying the cells, especially because we have different channels that you can see different dyes. I'm not going to get into what the different dyes mean because that's also different for each project, but as you can see here, currently for this image alone, we had quantified already around 250 cells, and we still weren't done with the manual counting of the cells in this image. So it is a lot of manual work, but that's science. So here you see some other memories from over the summer, counting the cells in the field of view and trying to stitch all of these positions, these microscopic positions, into one larger image, which unfortunately crashed my computer an inconceivable number of times. We don't talk about how many times I had to restart my computer. But the central message behind this, I guess, is that image processing is and will be a difficult task, both manually and conceptually, as you try to also develop a computer program to help you quantify cells or do whatever tasks that your project has you do. At this point, I'd like to call on my fellow intern Albert to see if he has anything to say.
Okay, well, one thing to note about Cellpost, which we did try to use, is like you can use this with Python as well as like Fiji. You can code your own macros for this. So if you want to like do some more coding stuff in your group, you can code your own macros, which would have made this counting cell process on Fiji MSJ a lot easier for us. And then I guess so using Python for Cellpost, you can load your own models and train your own models using Python instead of using the GUI provided. Like you can use that for I guess better analysis or more automated analysis. I guess that's about all I gotta say for now.
Thank you so much, Albert. And for just everyone's shared information, Albert was actually our group leader back during the summer. He did a lot of work behind the computer science and the image processing algorithms. As I mentioned before, I wasn't the computer science person, and walking into the program, I knew almost nothing about computer science. So Albert definitely did some hard carrying on the computer science side. So kudos to him for that. And that concludes my presentation for tonight. Go Opals, and I'd be happy to open the session up to some Q&A now.
Thank you so much, William, for that phenomenal presentation. William's just great. I can't even emphasize enough how highly motivated of a returned intern you are, William. Thank you so much.